Alright, so picking back up when we left off. Alright, so we're going to transition and from talking about the cell cycle, talking about the different stages of mitosis, what's happening in each. Alright, so again, kind of refocus on the cell cycle, kind of reconnect those points, what's about to take place in the cell. Alright, so here, looking at a cell that's an interphase. Alright, so a cell that's an interphase, alright, that's in G1. The nuclear envelope is intact. Alright. The chromosomes, so here we have 4 total chromosomes. Alright, so 4 total. That have not been replicated yet. Alright, so during the S phase, your chromosomes are replicated. Alright, so here during G2, your nuclear envelope is intact. Alright, and your chromosomes have duplicated. So that you have 4 chromosomes, but you have 8 chromatids. Okay. Alright, so you got 2 sister chromatids for chromosome 1, chromosome 2, 3, 4. Alright. Now, as the cell enters the mitotic phase, alright, during this process of mitosis, your chromosomes, they condense, they become highly compact, and we'll do a little hand waving right now, alright, because we'll talk about what goes on during each stage. But at the end of the mitotic phase, alright, at the end of mitosis, you have this process of cytokinesis where your individual daughter cells separate and form. And each daughter cell now has 4 individual chromosomes. Alright, so here you have 4 chromosomes, 8 chromatids, 4 chromosomes, 8 chromatids. Alright, so that means the total number of chromatids, genetic material, had to duplicate so that each daughter cell now gets the same complement of chromosomes, alright, that are identical to that of the parent in terms of the parent cell. Alright, so looking at mitosis, you can split mitosis up into a series of phases. Now it's important to note that we designate these phases with labels, and we talk about the important events that are happening in each stage. Alright, but you need to know that this is one big continuum. Alright, the cell doesn't go, hey, I'm entering prophase, now I'm entering prometaphase, now I'm going to enter metaphase. The cell makes this smooth transition, alright, through this mitotic phase, through mitosis. Alright, it's just that we have basically taken little snippets of time and looked at and compared what's going on throughout this entire process and designated labels to each of these different stages for the events that take place. Alright, so we have prophase, prometaphase, which is an intermediary step between prophase and metaphase, metaphase, anaphase, telophase. Cytokinesis is a completely separate process, but it does begin to occur during mitosis, during this latter step or stage of telophase. Alright, so order of steps occurs in this order. Alright, so a short little mnemonic device, PMAT, for prophase, prometaphase, metaphase, anaphase, telophase in terms of the sequence of steps in which mitosis occurs. Alright, so this kind of gives you a overall big picture of the process of mitosis. Alright, each individual step showing you the nice little continuum transition from one stage into the next. Alright, we're going to focus on what's going on during each individual stage. Alright, so here looking at an animal cell undergoing mitosis. Look at the individual chromosomes, which you can notice here. Alright, they're highly condensed at this point. Alright, they're kind of unorganized. Alright, they're being organized towards the center of the cell. Alright, so they all line up. Now they separate. So now your sister chromosomes are separated. They're now individual chromosomes. Now you have two independent daughter cells that are identical to the parent. Alright, so here, looking at the process of mitosis in a sea urchin. Alright, so right now you're looking at a sea urchin embryo. Alright, so you're going to be looking at different stages of development. Alright, following fertilization. Alright, so you have one cell division. You have two cell divisions. Now you have the formation of the little blastula. Alright, now you have a process of differentiation. You have different layers of tissues of cells that are forming. Alright, different layers of the sea urchin. Alright, so you have gastrulation, so you have this invagination of cells into the interior of the cell. Alright, it's going to form ultimately the digestive tract. Basically, in terms of gastrulation, it's a tube plan, alright, tube within a tube. Alright, so depending on whether you're a protostome, durastome, alright, that gastrulation, that invagination, is either first the mouth, right, for your protostomes, or the anus for your deuterostomes. Alright, so looking at this process of mitosis. Alright, so interphase. What goes on in the interphase? Well, cytoplasmic content doubles. Alright, your chromosomes are duplicated. Alright, your nuclear envelope is nice and compact. It's fully formed. You have your little nucleolus. Alright, a lot of times you'll see a little nucleolus present inside the cell. Alright, you can see your centrosomes. Your centrosome is referred to as your microtubule organization center. Alright, that is responsible for synthesizing your microtubule mitotic spindle fibers. Alright, so these microtubules are what attach to your chromosomes, and they attach at the kinetochore. Alright, so here you're looking at spindle fiber formation during mitosis. Alright, so you can see your spindle fibers elongating there. Alright, Alright, they polymerize and then they depolymerize as the cells begin to separate. Alright, so looking at prophase. Alright, so prophase at this point in time, your microtubules are beginning to be synthesized. Alright. Your chromosomes are going to condense. They're going to become more compact and more visible. Alright, during prometaphase, alright, your nuclear envelope is going to fragment. It's going to disappear. Your nucleolus disappears. At this point in time, your spindle fibers attach to the kinetochores on your chromosomes. Alright, so at this point in time, your chromosomes are going to be kind of all dispersed throughout the cell. They're not going to be organized in any way. Alright, as the spindle fibers attach to them, it's kind of a tug of war. Alright, your chromosomes are going to be pulled back and forth and back and forth. And eventually, your chromosomes are going to be pulled so that they all line up along this imaginary midline of the cell that we call the cell equator, the metaphase plate. Right, there's a lot of terms for it. The midline of the cell. Alright, so now all of your individual chromosomes line up. Alright, so you get chromosome 1, 2, 3, 4. Alright. And notice that each sister chromatid is facing one pole of the cell. Alright, so you have your east and your west, for instance. Alright, so that sister chromatid is facing this side, and this one's facing that side. Alright, so in metaphase, right? So now all of your little chromosomes have lined up in the middle, alright, along the metaphase plate. Alright, you'll notice here that your spindle fibers in yellow are attached to this black spherical structure on one side of the sister chromatid. Alright, that black spherical structure is a kinetochore. It's basically a protein complex for which the spindle fibers attach to. Alright, so these spindle fibers are being produced from your centrosome. Now you also have these shorter microtubule fragments that we call asters. Now these asters attach to the cell membrane as a point of anchorage. Alright, so these individual sister chromatids, alright, they're about to be pulled to opposite ends or poles of the cell. Alright, so that's what takes place during anaphase. Alright, so right now we're referring to each couplet, alright, each of these pairs of sister chromatids as an individual chromosome. Okay. During anaphase, your sister chromatids separate. At this point in time, each sister chromatid is referred to as an individual chromosome. Alright, so now we have a total of 8 individual chromosomes. Alright, and they separate. So during this separation, what's taking place? Well, if you'll recall that if you zero in and you look at your individual chromosome, alright, your sister chromatids are held together by your cohesin proteins. Alright, so these cohesin proteins have to be enzymatically cleaved. Alright, so there's an enzyme called separase that basically removes these cohesin proteins so that your sister chromatids can separate from one another. All right, and they begin to move towards opposite ends of the cell. All right, so here that's what's taking place. All right, your cohesin proteins are being separated, they're being cleaved, they're being removed, so that your individual sister chromatids separate. So now you end up with individual chromosomes. Alright, so looking at what's happening during this transition between metaphase and anaphase. Alright, so here, if you look at your little microtubule, alright, so this is your little spindle fiber. Alright, your spindle fiber, as it passes through the kinetochore, alright, which is basically one big protein complex, you have this motor protein that's basically ratcheting the chromosome forward along the microtubule. And as your microtubules at the other end of the kinetochore, they're actually depolymerizing. They're plucking off one another. Alright, so your little tubulin subunits that make up your microtubules are popping off. So in this case, your microtubules are in effect shortening. Alright, they're decreasing in length. Alright, as this motor protein walks the chromosome along the microtubules themselves are actually shrinking in length. Now, the last stage of mitosis is telophase. Alright, so during telophase, all the events of telophase are basically the complete opposite of prophase. Alright, so here, your nuclear envelope begins to reform. Your microtubule spindle fibers break down, your chromosomes relax, and you also have the beginning of the formation of cytokinesis. Alright, so here we're looking at an animal cell, so you have the beginnings of the formation of your cleavage furrow. Alright, so here you can look at two of the individual nuclei. So you get the formation of the nuclear envelope. Alright, so here you can see your individual nuclear envelopes beginning to reform towards the end of this process of mitosis and cytokinesis. Alright, so now you have more individual cells that are forming, being produced. Alright, so looking at this process of cytokinesis, alright, looking at animal and plant cells.
In animal cells, cytokinesis begins with the formation of a cleavage furrow. At the site of the furrow, a ring of microfilaments contracts, much like the pulling of drawstrings. The cell is pinched in two, creating two identical daughter cells. In plant cells, cytokinesis begins when vesicles containing cell wall material collect in the middle of the cell. The vesicles fuse, forming a large sac called the cell plate. The cell plate grows outward until its membrane fuses with the plasma membrane, separating the two daughter cells. The cell plate's contents join the parental cell wall. The result is two daughter cells, each bounded by its own continuous plasma membrane and cell wall.
Alright, so looking at this process of cytokinesis, looking at animal cells, plant cells. Alright, so animal cells, it involves the use of a contractile ring. Alright, this contractile ring is made up of microfilaments or actin. Okay, and as this actin microfilaments, this ring structure, as it begins to invaginate, it becomes tighter. Alright, it's very similar to tying a knot, or similar to basically closing around as a knot tightens. Right, that circle tightens and it becomes tighter and tighter on the cell. It basically pinches the cell in half. Alright, so here you have this physical separation of two animal cells at the end of this process of cytokinesis. Alright, two independent cells. Your plant cells, on the other hand, there's no contractile ring. You actually have these vesicles that are products of the Golgi body that begin to coalesce and form this cell plate. And this cell plate will eventually fuse with the pre-existing cell wall. New cell wall materials are laid down. Alright. And you have this physical separation between two independent daughter cells in terms of your plant cells.
This soccer player's knee will be as good as new in a few weeks, thanks to mitosis, a type of cell division that generates new cells for growth and repair. Let's move into a cell to witness the events of mitosis. Before a cell can divide, it must first duplicate the chromosomes stored in its nucleus. During chromosome duplication, several bubbles open up along the chromosome. Each bubble grows until it merges with an adjacent bubble. Each chromosome now consists of two identical copies called sister chromatids. Getting closer, we see that each sister chromatid consists of DNA wound around small proteins called histones. The sister chromatids begin to coil into tight helical fibers. Outside the nucleus, centrosomes that duplicated earlier move away from each other to opposite sides of the cell. Microtubules extend from the centrosomes, forming the mitotic spindle. Back in the nucleus, the DNA forms loops, becoming more compacted. These structures fold back on themselves, eventually condensing into a shorter and thicker chromosome consisting of two sister chromatids. As the chromosomes continue to condense, the nuclear envelope breaks up. The array of spindle microtubules is now extensive, and the chromosomes are fully condensed. Spindle fibers from each pole attach to protein structures located at the centromere of each sister chromatid. As the chromosomes are bound by spindle fibers from opposite poles, they move first one way and then another. The counteracting forces of the spindle eventually cause all the chromosomes to end up at the center of the cell, as if arranged on an imaginary plate. The sister chromatids are released from each other, each becoming a full-fledged chromosome. They are moved toward opposite poles of the cell, pulled along the spindle fibers attached to them. At the same time, overlapping spindle fibers that are not attached to chromosomes continue to lengthen, pushing the poles farther apart. Once the chromosomes arrive at their destination, they become less condensed. two new nuclear envelopes form, completing mitosis, the division of one nucleus into two genetically identical daughter nuclei. The cytoplasm divides by the process of cytokinesis, forming two separate daughter cells. In your body, millions of cells divide every second, providing new cells for growth and for repair of damaged cells.
Alright, so your prokaryotic cells. Alright, so your bacteria or archaea. They reproduce through a process referred to as binary fission. Alright, so in this case, your prokaryotic cells typically have a single circular double-stranded chromosome that's much smaller, encodes for less genetic material than your eukaryotic cells. Alright, so there are three main phases to binary fission. Alright, so first you have the replication of bacterial chromosome. You have the separation of the copies of your bacterial chromosomes. As this is taking place, the bacterial cell is elongating, and the copies of these chromosomes are moving towards opposite ends or poles of the cell, until eventually they're going to be physically divided into two new daughter cells. Alright, so here you're looking at the process of binary fission. You have your little bacterial cell, you have your single circular double-stranded DNA chromosome. That chromosome duplicates, so the genetic content has increased. You have elongation of the bacterial cell as this is taking place. You have invagination of the cell envelope, basically the cell wall and the cell membrane, in moving towards the cytoplasmic content. So invagination of the cell wall into the cell. Alright. This is going to form a physical separation between the two daughter cells. Alright, this involves a FITZ protein, a contractile ring, alright, where these two daughter cells are going to pinch off into two new cells. Alright, so this contractile ring structure is similar to, but not the same as, the actin contractile ring used in the formation of a cleavage furrow in animal cell cytokinesis. Alright, so here your two daughter cell bacterial cells are going to be separated from one another, and they have their own individual chromosome that is identical to that of the parent.